Hello, everyone. I'm Bojin. I'm a postdoc from KU I'm a part of FIBROTARGET Consortium. Today, I will bring you through how to read fibrosis and also how to use in vitro fibrosis model to evaluate the fibrosis in intestinal context. First, I would introduce what is intestinal fibrosis. So as we know during the inflammation, there's a epithelial barrier disruption. So gut microbiota can infiltrate into the tissue, and this will cause immune response. So T cell, dendritic cell, macrophage, lost immune cell will arrive into the damaged site, and this will cause for inflammation and also trigger the tissue repairing. So this means it will activate the fibroblast, and these fibroblasts will, in the end, repair the tissue for the regular wound healing. As you can see here in a regular colonoscopy, you can see during the regular tissue repairing, there is no scar tissue left. The lumen stay in a good architecture. But when there's a fibrosis, the scar tissue form and there's a narrowing of a lumen and it's causing the fibrosis. How do we identify fibrosis in human ileum? To answer this question, we will use H&E staining and Masson trichome staining to evaluate the fibrosis. So as here you can see, when the resection sample arrive in the pathology lab, we can embed it in the formalin, and then we can make a historical section, and then stain with hematocyanin and eosin. And by lead staining, we can evaluate inflammation, fibrosis, and alternation of tissue architecture. From the top panel, you can see in a healthy ileum tissue, the architecture remains intact. But when there's a fibrosis or inflammation, there's a ulceration in the mucosa side, and same apply for the stenotic tissue. And we can also use Masson trichome staining to label the collagen deposition. In this figure, you can see the collagen are being stained as a blue, and they are increasing when there's inflammation and fibrosis. So the next question will be, what are the cellular change in the Crohn's disease ileum? To answer this question, we will use a flow cytometry. So to do the flow cytometry, we need to get a single cell suspension. How do we achieve this? First, we will use a DGT and EDTA to break down the mucus and also release as much as epithelial cell as possible. And then further, we will use an enzyme to break down the collagen bound and to obtain the single cell suspension. Afterward, we will use antibody staining and acquire the antibody signal with a fluorescent detection by flow cytometry. This is an example how we identify the fibroblasts in the tissue. So after acquire the sample on the flow cytometry, we use a lineage marker, for example, CD45, CD31, and CD326 to remove the immunocell, endothelial cell, and epithelial cell. And then further, we can use a gating to find fibroblasts based on protoplanin and CD90 as a pan-fibroblast marker. And furthermore, we can use FAB and CD34 to identify the pathogenic subset, which is FAB fibroblasts in the Crohn's disease ileum. Next question is, how do we study fibrosis in vitro using primary human fibroblasts? In this section, I will bring you through how to isolate the primary human fibroblasts and different techniques to evaluate the fibrosis. So first of all, how do we isolate the primary human fibroblast? This is very simple. It will be exactly the same procedure as the cell suspension we obtained from the flow cytometry. And then we can culture them in the tissue flask and purify them and then identify with a vimentin. As you can see here, we confirm they are the fibroblasts. First, we will use a primary fibroblast to evaluate the fibrosis phenotype and also how the trigger can induce the cell behavior. In this section, we will use the live cell imaging so we can track the cell proliferation and also perform wound healing assay. In our lab or in this project, we use the InQSight system to follow the cell proliferation through different time points with a different cytokine stimulation. And of course, we can also perform the wound healing assay to understand the cell migration and also how long it will push the wound site closure. So next, we would like to also understand the pro-fibrotic phenotype after different stimulation using qPCR, Western blot, and collagen secretion. So from this slide, you can see here as example, we can use cytokines to stimulate and induce the collagen expression. And based on this, there's increasing of the collagen content by Western blot and the collagen content in the cell culture medium, and of course by qPCR. Next, to further understand more functional assay, we also can use in vitro generation of fibroblast-derived matrix. So in this assay, we mimic chronic inflammation. So we stimulate the cell multiple times to induce a chronic inflammation. So based on this, we can also apply the fibroblasts in different stage. And then we also supply the vitamin C to boost the extracellular matrix deposition in the in vitro setting. As you can see here, through different cycle of pro-fibrotic crew, and then also the inhibition, we can induce the matrix production. And this can allow us to evaluate if the compound can inhibit the fibrosis and what kind of cytokine can induce the fibrosis.
So I will follow up on my colleague Bojin, and I will introduce another in vitro model, 3D organoid models as a models of human intestinal tissue. My name is Veronika Bosáková, and I'm a postdoctoral researcher in a group of Professor Gianluca Matteoli at KU Leuven as a part of FIBROTARGET. So in this scheme, you can see different models that are used in the inflammatory bowel disease research. You can see the most complex in vivo models to the simplest, but the most high throughput in vitro 2D models. And each of the model has their own advantages and disadvantages. And based on your project and your hypothesis, you choose the one that fits the best. And in this part of the presentation, I will focus on the organoids that are 3D in vitro models of human tissue. So as I said, I will focus on organoids, and we have different kind of organoids based on the source of the cells we use to generate them with. So we can have patient organoids that are derived from adult stem cells. And in this case, the organoids contain only one specific cell lineage. In case of intestinal tissue, those are epithelial cells from the crypts of the human gut. But we can have also organoids that are derived from pluripotent stem cells or embryonic stem cells or induced pluripotent stem cells. All kind of organoids then we can use for gene editing, disease modeling, or interaction of different cell types, even with microbiome. And all of them we can use for high throughput screening, such as library screening or personalized medicine. In this part, I will focus on induced pluripotent stem cells, iPSCs-derived organoids, as this is a model that we use in our lab as a part of the project. So what are induced pluripotent stem cells? These cells are adult somatic cells that have been reprogrammed back into embryonic-like state, which gives them ability for self-renew and also gives them ability to differentiate into any cell type in the human body. They are made from the adult somatic stem cells that are introduced with transcription factors that are called Yamanaka factors. That gives the cells or reprogram the cells and erase their cellular identity and reset the epigenetic landscape, giving them ability to self-renew and differentiate to any cell types. Why we would use induced pluripotent stem cells instead of embryonic stem cells? It bypasses the ethical issues, and also the source of the cells is much easier to obtain, as we can use fibroblasts or peripheral blood cells. In the protocol for the differentiation of iPS-derived organoids, we follow organogenesis of the tissue, and we start with the stem cells, which we differentiate to endoderm, which is one of the three germ layers that are formed during gastrulation, and endoderm gives rise to key internal organs such as gut, liver, or lung. After the endoderm differentiation, we differentiate the cells to hindgut, which gives rise to major components of intestinal tract. And at this stage of the protocol, the cells form spontaneously clumps, which we call spheroids, that start floating in the media. And at this phase of the protocol, we collect the spheroids and we embed them in a extracellular matrix, which supports the cell growth. And after several days in the ECM, which can be matrigel, and supplementation with different growth factors that supports epithelial cell growth, we can see spontaneous formation of complex structures, which we then call organoids. With this model, we can perform different kind of analysis based on our hypothesis or goals in the project, and I will show you most of these analyses, how the results can look like. So I will start with the confocal microscopy and immunofluorescence staining, and you can see that the differentiated organoids, they form complex organized structures mimicking human villus-like structures. And also the organoids form functional tissue, and you can see production of mucins inside the lumen of the organoid. Most importantly, they also contain different cell types compared to patient-derived organoids. We have epithelial cells that form epithelial layer, but also the stromal cells here. And as my colleague Bojin mentioned in the beginning, epithelial layer during the inflammation is damaged, which supports the inflammation. And using organoid model, we can study also the epithelial layer and the damage or how the homeostasis. And here you can see that we can study tight junctions, which are crucial for the epithelial cells. As I mentioned, using organoids, we can focus both on the epithelial cells, but also on the stromal cells. And we can model intestinal inflammation using different combination of stimuli. And if we stimulate the organoids with pro-inflammatory cytokines, we can see damage of the epithelial cells, which is mark of intestinal inflammation. But we can also see activation of the stromal cells, which is mark of inflammation-induced fibrosis. Moving forward to different kind of analysis, which can be also RNA sequencing, we can look more on molecular level of the inflammation and development of fibrosis, and we can look at the gene transcription in the organoids. And you can see here that after stimulation with pro-inflammatory cytokine, here for example TNF, we can see a robust pro-inflammatory response in the mucosal tissue, marked by NF-kappa B activation. As I said, organoids consist of various cell types, but they do not consist any immune cells, which is a huge advantage for research of inflammatory bowel disease, as we can focus on specific immune cell subpopulation and study interaction of this subpopulation with with mucosal tissue without being biased by other immune cells. And other advantage of iPS-derived organoids is that they form so-called immunocompetent environment, which means that even though they do not consist any immune cells, they respond to different stimuli, and they can also interact with immune cells that were introduced to the system. Here you can see on the confocal microscopy picture monocytes in yellow interacting with the mucosal tissue of the organoid. With flow cytometry, we can then, as the same as in the tissue, we can focus on different cell types and describe their phenotype on the cell level. So in summary, I showed you that iPS-derived organoids and patient-derived organoids in general provide physiological and relevant model to study epithelial barrier, but also stromal compartment and fibrosis, and also interaction of different cell types that are crucial in inflammatory bowel disease. And by combining of this model and the state-of-the-art techniques, we can study the mechanisms and also test the different therapies for intestinal inflammation, but also fibrosis. And to summarize all the in vitro part, we showed you that we can study or characterize fibrosis in the human ileum using different histological staining methods such as hematoxylin and elzin or Mason trichome to study the architecture, but also collagen deposition. That Crohn's disease associated fibrosis involves different cell types, and there is a pronounced cellular heterogeneity, which can be characterized by flow cytometry. And then in the later part, we showed you different in vitro system, how we can study fibrosis development and how we can test different therapeutic strategies. And that's all from me. Thank you.